So let's go big picture first of all. With our big picture idea of what we're doing, let's start off by talking about the central dogma of genetics. We know that we start with DNA. In those little outlines you're using, what's the nickname the guy keeps using in the outlines for his DNA molecule? He calls it the $1 million blueprint. It's a valuable piece of building information. If you had something worth $1 million, where would you put it? It's safe. In your cell, where's it safe? In the nucleus. So DNA's stuck there. It's not leaving the safe. It's in the nucleus. But it's the directions for building proteins. And where does protein building actually take place? Outside the nucleus, at the ribosomes. Which, remember, they're not really organelles, but they're cell machines. So to get the information from the DNA to the ribosomes, we have to make a more portable and cheaper copy of the DNA, and that is RNA, messenger RNA. In the process of going from DNA to RNA, that process is called transcription, right? That's transcription. That's going to be our main focus today, transcription. Eventually, that mRNA will make it to the ribosomes. They're going to decode it, figure out what it's telling them to do, and actually build the proteins, assemble them. The process of actually building the proteins is translation. And then those proteins go on to do what? Make your traits. They determine your traits. And the one thing that we're leaving off of our diagram is our loop here. And our loop represents that DNA is constantly having to what? Replicate itself. And just a reminder, as far as we know, every living thing does this in this particular pattern. There's one exception that we know of, and it's not really alive. That exception is what? Retroviruses, which start out with what kind of genetic information? They're RNA viruses. So they can't start with DNA. So they're the exception. They've got a step in front. And what's the step in front called for them? Reverse transcription. They have to turn their RNA into DNA to start with. So let's talk about transcription. Before we get into transcription, though, he used the same diagram in the video we just watched. In eukaryotes, when replication is happening, it's happening at multiple places on a chromosome at the same time. They're different, what we call replication forks or bubbles, and they're all getting copied at the same time. What's that all about? Why do we need to be doing this at multiple places at the same time? How much DNA did we say you had in your body? A whole lot. And if you just had one little machine copying it all, how long would it take? It'd take forever. Right? So in eukaryotes, you've got what we call multiple replication forks. They're all copying on the same chromosome, and eventually all those pieces will get put together. In a bacteria, it works a little bit different. How many chromosomes in a general bacteria cell? Just one. So in a bacteria, turns out there's only one replication fork. Remember that bacterial chromosomes are circular, right? So there's no need to have multiple replication forks. It just starts in one spot, copies it, and you end up with two loops. So point there, eukaryotic cells, like us, have multiple what we call replication forks or replication bubbles, so that our DNA can be copied fast. It's how many chromosomes are in a typical human cell? 40 what? 46, and they're big, they're long. In a typical prokaryotic cell, there's only one, and it's just a short loop, right? So speed is not an issue with bacteria. With eukaryotes, it's an issue. You need to be copying these things multiple times very quickly. Before we get into the guts of transcription, we need to know a little bit more about RNA. Turns out there's several kinds of RNA. More than what we have up here. But these are the only three we're going to be concerned about. M, T, and R. Before we go further, let's make sure we know what the M, the T, and the R stand for. The M is the one you all probably know about. It's messenger RNA. Its job is to be a messenger between what two things? It's a go-between between the DNA and the ribosomes. It's the messenger between the DNA and the ribosomes. But the tRNA you may not know about. You may not have talked that much about tRNA in ninth grade biology. Does anyone know what the T stands for in tRNA? Speak up. Transfer. Exactly, transfer. I want you to think of tRNA, the T. Instead of transfer, I want you to think of it like taxi. Taxi. Because what's tRNA's job? Anybody know? We haven't talked about this yet. Its job is to transport around what? What does it deliver to the right spot? You may not know this. It's a transport for amino acids. It takes the amino acids and puts them where they're supposed to go. So think of it like taxi RNA. It's a taxi for amino acids. By the way, where do you get the amino acids that you use to build proteins in your body? Where do you get your amino acids? From food. That's why it's really important that you eat a balanced diet. Who has the most problems with protein deficiencies? What group of people? Other than poor, undernourished people. Vegetarians. Vegetables, fruits, have proteins in them, but they don't always have what we call complete proteins. What do you think a complete protein means? It means it's got all the different what? Amino acids. Not all vegetables have all 20 amino acids. If you're a vegetarian, you can get all the protein you need. I'm not saying don't be a vegetarian because it's healthy. If I could be a vegetarian and have a full belly and feel good about it, it'd be the way to go. But I need to eat a little bit. It's just the way I am. But if you're a vegetarian, you need to mix certain foods together. For example, all over the world, regardless of culture, a lot of people have mixed two vegetable dishes all over the world. Beans and what? Rice. And guess what you get when you put beans and rice together? All 20 amino acids. It's a balanced diet. Whether we did that by accident, how we figured that out, way before we knew about DNA, I don't really know. That's a good combination. So vegetarians, what I'm saying is they have to watch what they eat. They have to eat certain foods in combination to get the 20 amino acids they need. If you eat a steak, guess what you get? You get all 20 amino acids. But you also get what? You get cholesterol, fat, and heart attacks, okay? It tastes good, okay? But you also get the side effects. Yeah, you gotta live a little bit. All right, the last type of RNA, the rRNA. What does the R and the rRNA stand for? You guys may not know this. It stands for ribosomal. Because by far, the ribosomes, the biggest part of a ribosome is actually made of RNA. So rRNA makes up ribosomes. That's what they're made out of. rRNA. Where in a cell do ribosomes get put together? Where do they get built? There you go. They get built in the nucleolus of your cells. That's what that nucleolus is all about, is building ribosomes. Remember the nucleolus, if this were a eukaryotic cell, and this was the plasma membrane, and this was the cell membrane, most eukaryotic cells have a real dark spot in their nucleus. That's the nucleolus. And what's going on there, that's where ribosomes are actually being made. Now, do they stay there? No, they get transported out here, and where do most of them end up? Attached to your rough ER, right? So they get exported out of the nucleus after they're made, but they do get made there. They start out in the nucleus. Nucleolus is where ribosomes are made. Now I'm going to go to a picture to talk about this other stuff. Maybe not. I have a markers out of spot. All right, here's a picture of RNA. We want to make sure we know some differences between RNA and DNA. Number one, let's talk about some similarities first. They're both what kind of chemical? Nucleic acids, which means they're long chains of what? Long chains of nucleotides. Nucleotide, 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 nucleotide. They're all nucleotides. But some differences now. Number one, what do we notice about the number of strands here in RNA? It's just one. It's a single helix instead of double helix. One helix. Also notice the sugar, you can't really see a whole lot of differences right now in this picture, but the sugar is different. The sugar, instead of being deoxyribose, is ribose, which means that sugar just has one extra leg, one extra oxygen atom. Doesn't make that big a difference, but that's just the way it works. And then the other difference that you ought to note is that one of the nitrogenous bases is slightly different. Whenever we would have a T in DNA, we end up with a uracil in RNA. So we have uracil instead of thymine. So the three main differences: single helix instead of double, ribose instead of deoxyribose, and uracil instead of thymine. Make sure you know those three differences. Those are important. All right, now let's talk about transcription. When we were going through replication, we went through lots of enzymes. And you got to know all those enzymes, because the people that write the AP exam want you to know all those enzymes. Transcription, there are several enzymes involved here too. But typically the only one that they really want you to know is the main one, and that's called RNA polymerase. Sometimes it's called RNA polymerase 2. RNA polymerase, it's the main enzyme of transcription. Now there's a couple of things you got to be careful of. Let me put up a little strand of DNA here. All right, let's go ahead and put some numbers on here. Let's say this is our 5' end. This is our 3, which means this is going to be a 5, and this is going to be a 3. All right, everything in black up here is going to be DNA. Everything in green up here is going to be, guess what? RNA. First thing that happens during transcription is that these two strands have to be what? Unzipped. RNA polymerase does that itself. Helicase is not involved in this process. RNA polymerase unzips the DNA. This is where things get different though. RNA polymerase, just like DNA polymerase, can only work from 5 to 3. But in DNA polymerase, how many of those chains did we copy? We copied both of these during replication, right? In transcription, it turns out we're only going to copy how many? We're just going to copy one. Anyone know what that strand is that we use to make RNA? What's it called? No. It turns out to be the leading strand, but it's got a name because of what its function is in transcription. It's called the template strand. The template strand, or sometimes you might also see it called the sense strand. Well, if we're reading from bottom up, this is 3 to 5, which means we're going to be building from 5 to 3. So it would be this one, right? This would be our sense strand if we're reading from the bottom up. So this gets unzipped. Remember, we're still connected down here, still connected up here. We're working in a bubble, in this case a transcription bubble. RNA polymerase sees a C here. What's it going to stick to it? A G nucleotide. And remember, it's not just the G. What's always connected to that G? It's a nucleotide. So it's a nitrogenous base plus phosphate plus sugar. I'm just showing you the nucleotides, the nitrogenous bases, but the other stuff's there. C's a T, what's it stick in? An A. It C's a T, sticks in an A. C's a C, sticks in a G. C's an A, sticks in a U. Remember, no T's. Wherever there should be a T, we put a U, a uracil. C's a G, puts in a C, A puts in a U, and eventually it gets to a code that tells it to what? Stop. So back here somewhere, there's a code that says, hey, start here. This is the beginning of a what? What would this be? What are we trying to do here? We're trying to make a protein. What's a part of DNA that codes for one protein called? A little piece of DNA that codes for one protein. Let's flip the word for that. A gene, okay? So you've got a signal down here that says, hey, this is the beginning of the gene. Start here. And then you've got a signal up here that says, hey, end of the gene. Stop. When it stops, what happens to this little green piece of RNA? It breaks off. What happens to the two strands of DNA? They wind back up. They go back together. So the DNA is not changed. The RNA comes out. And that piece of RNA that we initially get is called the primary transcript. Now here's something that's a little confusing. If you're not paying attention, this is going to confuse you quite a bit. In bacteria, that's the end of transcription. They're done. That RNA is now going to move to where? Ribosomes. Ribosomes are going to decode it, and it's going to be used to make what? Protein. And that's it in bacteria. We've discovered in the last few years that there's another step in eukaryotes. Turns out to be a really important step. It hasn't been understood for that long. For years, scientists thought that we had a bunch of DNA that they referred to as junk DNA. They knew that we had a lot of DNA that didn't seem to be doing anything. For example, let's start to clean off the board here. They knew that we had long sequences of DNA, and a lot of times it seemed to be repeated pieces. Where this didn't actually seem to do anything. That they knew this was the DNA, but when they saw the RNA that was made from this, they saw this. So this never got what? It doesn't look like it ever got transcribed. It looks like it got left out. This part never was used in making RNA, they thought. And what they realized is it does get used to make RNA, the primary transcript. So what we would initially get, let's say this is our 3 to 5 strand up here. This is our template. What we would initially get would be this. Then in eukaryotes, there's another step, and that other step is called processing, RNA processing. What happens, depending on what's going on in your cell, certain pieces of this RNA get taken out before what? Before translation. Which means each gene doesn't just code for one protein. Because depending on what they take out, what do you get? I mean, if we took out this, that would lead to one protein, right? But if we took out this, that would lead to something else, right? If we took out this, that would lead to something else. So what scientists have found is that we have these pieces of DNA called exons and introns. Bacteria don't have this. In bacteria, all their DNA is used to make working RNA. In us, the parts of the DNA that are actually transcribed and translated, those are called exons. That's for years what they called the good DNA. And for years they called this, guess what? Well, you call it the junk DNA. Because they didn't really understand what was going on. But what they realize now is that what's an intron one time through, what might happen the next time through? It might be part of an exon. So that one gene lets us code for what? Many proteins. When they started doing this Human Genome Project back a few years ago, they figured out that, hey, we don't have as many genes as we should. I thought, wow, We thought we were going to have millions of genes, or at least thousands and thousands. And then they figured out we don't have that many. And the reason we don't have to have as many genes as they thought we would is that each gene is actually coding for what? Many proteins, because of this intron-exon thing. Exons are the part that are actually translated. Introns are the part that are taken out, removed, during, what's the step called when we're messing with the RNA? Processing, during the processing step. Remember that in eukaryotic cells, processing happens between what and what? Transcription, translation. And you could really screw up a piece of DNA here if you didn't watch it. Because if the wrong pieces are removed, what's going to happen? You're going to get the wrong protein. So it depends on your body. There's a lot of really crazy cell signaling going on in your body to say, hey, this needs to be taken out, this needs to be left. This needs to be an intron, this needs to be an exon. So there's all kinds of hormonal, chemical signaling going on to tell your body what needs to be taken out and what needs to be kept. Yeah. Introns are taken out, exons are translated. All right, now a word that you need to recognize. There are these enzymes in your body called spliceosomes. What's it mean to splice? It means to cut and then what? Put back together. Like when teachers used to use old-timey film strips. You guys ever seen an old-timey film strip? The old film strip projectors? You see those things would get all bent up sometimes. And what you have to do is you have to cut them, cut out maybe one frame, and then tape together the other frames. That was splicing. These enzyme spliceosomes cut out what? They cut out the introns and then put back together the exons. Make sure you know that. For some reason, the people who write our AP exam, they like that word spliceosome and they like to ask questions about it. Spliceosomes are the enzymes that carry out RNA processing. They take out the introns, splice together the exons. And that's what we call process. Remember, that happens in eukaryotes, but not what? Not in bacteria. So here's the key thing, and we'll get more into this in the next few weeks. If I wanted to take a piece of human DNA and put it into a bacteria, which it turns out happens a lot. Scientists do that intentionally. What's maybe the best example of putting human DNA into bacteria? The most useful example that we've done to this point. Anybody know? Anybody in here diabetic? No? What do diabetics need to take every day? Insulin, which is, guess what? What is insulin? It's a what? What kind of chemical is insulin? It's a protein hormone, which means it's coded for by DNA, genes. So scientists found in the human genome, in the human DNA, hey, here's a gene for insulin. And what they did is they cut that out, and then they put it into bacterial DNA, spliced it into a bacterial chromosome, put that into bacteria. When the bacteria reproduced, what did they do to that human gene? They copied it. Every time they copied their own, they copied the human one too. How long does it take a bacteria to do that? Like 20 minutes. So if you make one bacteria that way, by the end of the day, you've got millions. And what are they all chugging out? It's insulin. That's how they make insulin for diabetics. It's human insulin made in what? Bacteria. They grow it in big, like room-sized vats filled with bacteria food, basically. And they process it. And it's human insulin. It's just like yours. Before they figured out how to do this, where'd the insulin for diabetics come from? Before they figured out how to make it genetically like that. Maybe from other people, but usually not. Usually they got it from either cows or pigs. Take it in. Thank you, sir. Now there's two problems with that. Number one, if it's cow or pig insulin, what do we know about cow DNA and pig DNA and human DNA? Are they the same? No. They're a lot the same, right? Probably like 85-90% the same. But is their insulin protein going to be exactly the same as your insulin protein? Nope. Which means it's not going to work. It's not going to work as well. It'll work, but not as well. So that was a problem. And also, there was always a worry about getting what? if you're taking insulin from a cow, there's always this risk of getting germs from the cow or the pig. This way, it's human DNA, it's human insulin, it's good. And that's the way they make it all the time. But before that would work, what do they have to do to that human DNA to put it in the bacteria? Take it out away. Introns, because bacteria don't know how to do that, right? They don't have the spliceosomes. They don't have the machinery to do RNA processing. They're not capable of that. So before you put a human gene or any kind of eukaryotic gene into a bacteria, the intron sequences have to be taken out. And you have to put only in the good DNA, the exons. I'm struggling here, guys. All right, anyway, let's move forward just a little bit. All right, transcription happens in 3 phases. And you need to know these 3 phases of transcription. It turns out they're the same 3 phases of translation. Transcription and translation both happen in 3 parts. The first part in both processes is called initiation. That makes sense. Start. And remember, this PowerPoint is posted on the website already, so you don't have to write all this down unless you just want to. But in initiation, there's a protein called a transcription factor that attaches to a part of the gene called the Tada box. Now that sounds like a weird name, Tada box. What do you think that means? There's a lot of right before the beginning of the gene. T's and A's. There's a lot of thymines and a lot of adenines, okay? So just before the gene, there's a long sequence of DNA, T, A, T, A, T, A, T, A. And that's basically saying, start here. This is where the good stuff happens. This is the gene. This is the beginning of the gene. Start here. So the Tada box is the signal to say, hey, here's the gene. Start here. Sometimes the Tada box is called the promoter sequence. It's a place to start. All right, and like it says here, it says this orients the factory to the direction that it's going to transcribe. It tells RNA polymerase, the transcribing enzyme, start here, go in this direction. What now? What? With the TA up there? I don't think so.
Sounds initiation.
All right, there's also some other proteins involved to get this started. Together, they're all called transcription factors. But the main protein you need to know the name of is RNA polymerase. It's the enzyme that actually does the work. RNA polymerase. RNA polymerase 2. Once this joins, it starts building RNA. And it builds it in which direction? 5 to 3, just like DNA polymerase. What you can see, though, from what we're talking about here is this is a pretty complicated process. It's controlled step by step in a very precise manner. Because if it wasn't, you'd have proteins all over the place that you didn't need. You'd be wasting energy. Your cells would kill themselves with waste of energy or waste proteins. So it's very controlled. There's all kinds of cell signals that control this process. The second part of transcription is elongation. So you got initiation, where RNA polymerase and the transcription factors bind to the promoter area. Then you've got elongation. And this is where the chain is actually being built. This is the building of the RNA chain. And remember that the way it works is RNA polymerase 2 unzips the DNA. It only uses how many strands to build RNA? Just one, and we call that strand the template or the sense strand. Builds it from 5 to 3. Template or sense. S-E-N-S-E, sense strand. Because it makes sense to the RNA. Now, a word in the outlines that I haven't mentioned. This word right here. Nucleosides. I haven't used that word. I've been using nucleotide. In your nucleus, you're going to have a lot of spare parts for making DNA and RNA. Those spare parts I've been calling nucleotides. But they're really nucleosides. So what do they have attached to them that a nucleotide does? Anybody pick that up in the outline yet? The outline posted up yet? Nucleosides have a couple of extra phosphate groups attached to them. which means they have a little bit of extra weight, energy. So what's going to happen, to make a new bond, what's required? Energy. Okay, so those phosphates are going to help provide the energy to put these nucleotides together. So just realize that if you see that word nucleoside, it's the same thing as a nucleotide. It's just a nucleotide with a little extra weight. Energy so that it can bind and make the chain. Don't overcomplicate the terms here. Nucleotide, nucleoside, you're going to hear me use them interchangeably to some extent. But these are in the cytoplasm. We get them from our food. That's where they come from. We get them from our food. After we break down food with our lysosomes, we reuse them. So elongation is building the chain, building the RNA chain. And then the last thing is termination. Terminate just means what? Stop. So that's just what it means, stop. Eventually, RNA polymerase gets to a sequence of codes, a codon, and that codon just says what? Stop. Stop here. What you doing? You're at the end of what? You're at the end of the gene, stop here. That's what termination means. You're at the end of the gene, stop here. Because this is a chromosome we're talking about, right? How many genes are on a chromosome? A bunch. There's no set number, a bunch, right? So it's got to stop. It gets to the end of a gene, it needs to know, hey, stop here. It may not need to make the next protein, it may only need to make just that one. So make the right protein, stop. All right, the last thing we'll talk about today is this right here, and we sort of already talked about it. In between transcription and translation, we have the processing thing going on. This sort of walks us through the idea of processing. Prokaryotic versus eukaryotic. In prokaryotic DNA, there basically is no processing. The RNA that's made is the RNA that's what? Translated, used to make proteins. There's no processing going on. But in eukaryotes, there's several things that gets done to the original piece of RNA that's made. First thing that happens is something called front-end modification, 5' modification. And this is something we haven't talked about. To the front of that new piece of RNA, there's what's called a 5' cap put on it. Some nucleotides put on the front to protect it, to keep it from breaking down. Like it says here, this is sort of like putting hard hat on your RNA to keep it from getting broken apart. It also is a signal that helps it attach up to the ribosomes. So this 5' cap helps it bind to the ribosomes and attach. To the back end of the RNA, the 3' end, there's what's called a tail put on it, a poly-A tail, which means we put a long chain of what on the end? A's, adenines.
You said it was bond to what?
So what else? So to bind to the ribosomes. So at the back end, we put on a lot of adenines, 50 to 250 you put on the back end. And again, this helps to protect it from being broken down. And it also helps to tell it, hey, we're done here. This is the end of the chain, the end of the gene. So the first thing that happens in eukaryotic DNA is we add a cap on one end and a tail on the other. And then finally, what we call middle modification. And this is what we were talking about earlier, the middle modification. We need to take out what? What needs to be taken out of the introns, the non-coding sequences, the sequences that don't need to be used at that point. And remember that the enzymes that do that are called spliceosomes. You're going to sometimes see them abbreviated as SNRNPS. Sometimes you're going to hear them called SNRPs. SNRPS, SNRPs, and splice. We're going to use those two words interchangeably, SNRPs and spliceosomes. But what you need to realize is those are the pieces of enzyme that take out the non-coding introns and then splice back together the coding exons. And that's a good place to stop on this for right now. Questions? All right, some things to talk about before we go any further. On Wednesday, we're going to finish up the translation stuff. Pretty much finish this DNA stuff on Wednesday. Hopefully, we're going to do a little lab activity on Wednesday. It's going to be more a paper and pencil kind of lab. But you're going to walk through replication, transcription, translation with a bunch of gene segments. Won't actually be any chemicals involved in the lab activity, just more of a paper pencil lab activity. After we finish that up, our next topic is mitosis meiosis. And when we get to there, we'll have several labs where we go in and use the microscopes to look at actual cell division. Let me turn off my recording first.